Welcome back to G Fun Facts Online. Today we are diving deep into a subject that sits right at the intersection of thermodynamics, biology, and the future of human survival. We are talking about the quest to stop biological time. Specifically, we are looking at how a revolution in the physics of freezing might finally solve one of the biggest logistical nightmares in modern medicine, which is the organ transplant bottleneck. If you look at the current state of organ transplantation, it is essentially a high-stakes race against a relentless clock. From the exact second a heart, liver, or kidney is recovered from a donor, a biological countdown begins. For a human heart, that clock is brutally short, offering a mere 4 to 6 hours of viability. For a liver, you might get 12 hours. This time constraint is the reason why thousands of perfectly viable organs are discarded every single year. They simply cannot reach a matched recipient in time. A heart donated in New York often cannot save a patient in California, let alone New Zealand. For decades, scientists have dreamed of a solution that sounds like it was ripped from the pages of science fiction. The goal is to suspend life indefinitely. If we could freeze organs and store them in banks, just as we do with blood or embryos, the waiting list for transplants could virtually disappear. But biology has a nemesis, and that nemesis is ice. To understand the solution we are discussing today, which is called isochoric vitrification, we first have to understand why freezing is so deadly to living tissue. When water freezes, it expands. We all know this from leaving a soda can in the freezer. But on a microscopic level, that expansion forms jagged crystals that act like millions of microscopic daggers. These crystals shred delicate cell membranes and turn functional tissue into mush. To stop this, scientists developed a process called vitrification. Vitrification is a method that turns biological water into a glass-like solid without crystallizing. It is essentially flash freezing so fast and with such a specific chemical mixture that the water molecules don't have time to arrange themselves into ice crystals. Instead, they get stuck in place as an amorphous solid, like glass. This sounds perfect, but traditional vitrification requires toxic levels of antifreeze, known as cryoprotectants. This created a seemingly unsolvable puzzle for cryobiologists for the last 50 years. On one hand, freezing kills via ice formation. On the other hand, vitrification kills via chemical toxicity. It seemed like a lose-lose situation. But recently, a quiet revolution in thermodynamics has begun flipping the script. It is called isochoric, or constant volume, preservation. By simply locking biology in a rigid metal box, researchers are bending the laws of physics to solve this puzzle. To understand why isochoric preservation is so revolutionary, we must first look at how we have been freezing things for the last 5,000 years, from the ice houses of ancient Rome to the modern kitchen freezer. Almost every freezing method in human history is isobaric, meaning constant pressure. You put an object in cold air at 1 atmosphere of pressure, and as it cools, it shrinks. When the water inside tries to freeze, it expands. Because the air around it yields, the water expands freely, crystallizing into ice and destroying the tissue. Isochoric preservation changes the fundamental boundary condition of this equation. Instead of air, the organ is immersed in a liquid solution inside a rigid, thick-walled chamber, effectively a high-tech, indestructible metal jar. The container is sealed tight with absolutely zero air inside. As the temperature drops, the water in the solution wants to freeze and expand. But the rigid walls of the container will not budge. This creates a thermodynamic standoff. The water pushes out, the walls push back, and the pressure inside the chamber skyrockets. Here is where the magic happens. Thermodynamics dictates that pressure depresses the freezing point of water. As the pressure rises, the temperature required to form ice drops lower and lower. This allows the system to reach sub-zero temperatures without the water inside the organ actually freezing. The thermodynamics of the universe are essentially tricked into keeping the water liquid or glassy at temperatures where it should be solid ice. This metal jar technology has spawned three distinct approaches, each tackling a different part of the preservation puzzle. The first mode is called isochoric freezing, or equilibrium. In this mode, a small amount of ice is allowed to form, but crucially, it forms in the solution surrounding the organ, not inside the organ itself. This ice formation drives up the pressure, which then prevents the rest of the water from freezing. The organ sits safely in a supercooled liquid pocket, protected by the pressure generated by the sacrificial ice on the periphery. This method is already showing immense promise in the food industry, preserving the texture of cherries and potatoes perfectly, and serves as the foundational concept for organ storage. The second mode is called isochoric supercooling, and this is the current champion of the field. This is where the most tangible recent successes have occurred. In groundbreaking experiments led by researchers like Dr. Boris Rubinsky at UC Berkeley, whole pig livers have been kept in a supercooled state. This means they remain liquid, but are below freezing, sitting around -4 to -6 degrees Celsius, without any ice formation for up to 48 hours. In a traditional cooler, a liver at -6 degrees would freeze solid. But inside the isochoric chamber, the pressure stabilizes the liquid state. This extends the metabolic suppression of the organ, slowing its decay significantly more than standard refrigeration. In recent trials, these supercooled livers were successfully transplanted into pigs and functioned immediately upon rewarming. That is a massive leap forward from the standard 12-hour limit we see today. But the third mode is the true holy grail. It is called isochoric vitrification. While supercooling buys us days, vitrification could buy us centuries. As we mentioned, vitrification turns the organ into a glass where molecular motion stops completely. In traditional isobaric vitrification, you need to pump the organ full of cryoprotectants at extremely high concentrations to prevent ice. That is essentially embalming the organ in toxic syrup. It preserves the structure, but kills the cells via chemical toxicity. Isochoric vitrification proposes a brilliant workaround. It uses pressure to lower the chemical requirement. Because high pressure naturally suppresses ice formation, you technically should not need as much chemical antifreeze to achieve the glass state. If isochoric confinement can reduce the toxicity threshold by even 20 or 30%, it could turn lethal vitrification into a survivable suspended animation. However, science is rarely a straight line, and isochoric vitrification is currently the subject of a fascinating intellectual debate. On one side, proponents argue that the synergy of pressure and rapid cooling in a confined volume facilitates glass formation with far lower toxicity. They point to successful experiments with coral fragments, which were isochorically vitrified and revived. This is a critical breakthrough for conservation biology, given the state of dying coral reefs globally. On the other side, thermodynamic modelers, such as the group led by Dr. Yod Rabin at Carnegie Mellon, have raised a red flag. They argue that cryoprotectant fluids contract significantly as they cool. This is known as thermal contraction. They suggest that the pressure inside the rigid box might actually drop instead of rising because the fluid shrinks away from the walls. If the fluid shrinks, you lose the pressure benefit, and you might even create a vacuum or cavitation that could damage the tissue. This scientific battle is driving rapid innovation. Researchers are now developing smart chambers with active plungers or flexible compensators to maintain the pressure sweet spot, ensuring that the contraction does not ruin the isochoric effect. One of the biggest challenges in this field was simply seeing what was happening inside the box. You cannot look inside a solid steel block to see if an organ has frozen or vitrified. To solve this, engineers developed the isovitroscope. This is a specialized chamber equipped with sapphire windows and high-speed cameras, capable of withstanding tens of thousands of pounds of pressure. This device has allowed scientists to watch the dance of ice and glass in real time, observing how crystals nucleate under pressure and how the glassy state sweeps across a sample. It is essentially the Hubble telescope of cryobiology, revealing a world of high-pressure physics that was previously only theoretical. The implications of solving this puzzle are staggering. The most obvious benefit is the end of waiting lists. If we can bank organs, every patient could receive a perfectly matched organ, virtually eliminating rejection. But the logistics go further. A kidney could be flown from Boston to Bangalore without rushing. Beyond organs, this technology could preserve limbs for reattachment or complex bioengineered tissues. And then there is the final frontier, space travel. NASA is actively funding isochoric research. If we are to send humans to Mars, we need a way to store food and potentially medical biologicals for years with minimal energy. Isochoric storage is incredibly energy efficient because you do not need to actively pump heat out once the equilibrium is reached. The physics does the work for you. We are standing on the precipice of a new era in medicine. For 50 years, we accepted that biological time was absolute. We accepted that once blood stops flowing, the clock ticks down to zero. Isochoric preservation, and specifically the pursuit of isochoric vitrification, challenges that inevitable decay. By confining life in a constant volume, we are learning to hit the pause button. While the perfect, non-toxic vitrification of a human heart remains a few years away, the path is no longer blocked by the laws of nature. It is being paved, one steel chamber at a time.